Thank you so much, I know, and thanks to all of you for joining. What I'd like to do is tell you a little bit about some research we published last year where we had a really innovative study design that allowed us to uncover connections between the diet, the host, and the gut microbiota and energy balance. And within that design, we were then able to start exploring the role of various metabolites, both in a targeted and untargeted manner. So there's two parts to my talk. In the first part, I'll give you some framework. It's kind of my idea of a thought experiment, get everybody kind of getting excited about the gut microbiota and some of the nuances. Then I will guide us through the gut microbiota and energy balance and how those two things connect. Then I'll spend a little time on exactly how we conducted the clinical trial, some of the key methods that we think gave us the precision necessary and the level of control necessary to achieve significant results with a small sample size. And then I'll finish this part with a few of the top line results. There are many more results in this study, so I'm hoping during the Q&A you might be curious about some of those, and I'd be more than happy to dive into those. So I've been a clinical dietitian for about 26 years, and I can tell you that for most of my career, I have not thought very much about bacteria, except in the context of food poisoning, because as dietitians, we get trained about cooking temperatures and avoiding harmful bacteria. But clearly for me and for a lot of you, this has been transformed by the knowledge that these little microbes that live in and on our bodies have a huge impact on our physiology. So also, mostly through my training in nutrition, before I really started thinking about the gut microbiota, I really envisioned, and I think most nutrition people envision the colon as this inert repository where whatever food that hasn't already been digested and absorbed in the small intestine and it's performing its magic on the human, it just sort of lands there and sits around until it comes out the other end. But now knowing the community of microbes that reside in the colon, I think of the colon as the human colonic bioreactor. So it's an active dynamic environment where multiple reactions are going on due to the activity of these microbial communities. And what's so important about our bioreactor is that the reactions just don't remain contained within. They transverse the gut lining. Signals from the microbes can speak to pretty much every single tissue. And there's this back and forth communication between the host and the microbiota that's impacted quite dramatically, actually, by diet. And in this back and forth communication, one of the major outputs of microbes are metabolites. And that's why I think it's so exciting to have tools to be able to dive into the metabolites. The other sort of interesting aspect of the community of microbes in the colon is that we mostly think about bacteria. I'm speaking today essentially about bacteria only, but there are also viruses, yeast, and fungi that are also in those communities, and they also have biological roles. So it's a very complex network that we're still looking to better understand. So with all these physiological connections, it's not surprising that you could go to PubMed and look up the disease that's most important to you, and you will find dozens, hundreds, maybe thousands of publications associating gut microbiota composition or maybe their metabolites with various disease states. But the big question is, are these associations causal, or are they just a reflection of the health of the host? And more importantly, how does this all work anyway? How do we figure out these connections? So I basically redrew and very much simplified a beautiful graphic from this Nature Reviews microbiology review by Fan and Peterson that I have at the bottom there, breaking it down into some layers just to contextualize how this all works and where the metabolites play a role. So you have to start with an input, right? If you have a bioreactor, you have an input. And the primary and perhaps the most fundamental input is diet. We all eat every day, and the microbes depend on us to feed them. There are also some host metabolites that could be inputs, such as primary bile acids, and a few limited metabolites that cross from the circulation back into the colon. Then those inputs hit the bacterial communities. All of you in this virtual room have your own unique community. The size of the community, the diversity of the community, the function of the community varies. And once your community is exposed to these inputs, then the microbes make an output. Many of them are metabolites, also antimicrobial peptides, and even the surface molecules that some of the microbes carry have biological activity. Then these outputs interact with the gut lining in one of multiple ways, either a direct receptor-ligand relationship, some sort of transport, or activation of a signaling cascade within the enteroendocrine cells that releases a product. These products then enter host circulation, metabolites being one of them, but also hormones like GLP-1 and cytokines, inflammatory molecules, and finally all that comes together to give an output. Of course, this is simplified to suggest a very linear relationship, but we know all these things are talking back and forth, adding to the wonderful complexity of the system. So I hope now you're thinking, okay, well, maybe this isn't just another gut microbiota presentation. Maybe we need to be thinking about the microbes in a different way. And one of the ways that we obsess about the gut microbiota at the Advent Health Translational Research Institute is energy balance and how that all impacts obesity. So I'll give you a few points here to consider. First of all, energy balance is energy in and equaling energy out. Most of the energy in for us is any dietary component, food or beverage. Energy out, I think, is where it gets kind of super interesting because there are many pathways to utilize that energy expenditure, exercise, fecal energy loss, which will be a particular focus for us, and then perhaps other mechanisms that we don't quite understand. So a calorie is a calorie when it goes in, but the utilization can vary between people. So one of the most fundamental sets of discoveries that caught my attention about the gut microbiota is its relationship with obesity. And this is a quick flyover of a few high-level points of what the literature suggests so far. A really nice review at the bottom that you might want to check out if interested. So in humans, either the relative abundance and or differences in microbially derived metabolites can differ in the lean versus obese phenotype. They can be predictors of weight loss response, or there can be differences in response to weight loss. But how this happens is what we don't fully understand. There is some evidence, mainly preclinical, but the human data is emerging, suggesting the gut microbiota can impact how much of that sort of leftover energy that hits the bioreactor is ultimately available to us, because it can go two different ways. How the microbes impact satiety and energy intake, and potentially also energy expenditure. So we were very intrigued by all the literature coming out about the gut microbiota and obesity. And we thought we had a way to really address some of the fundamental gaps in the field. The major gap is that we don't quite know in humans, although in preclinical models it's fairly clear, whether the gut microbiota is on the causal chain for the development of obesity, or if it's merely a reflection of host weight status. Or could it be both? And most likely it's both. So to address this fundamental gap, we set out to answer a fundamental question. And my favorite thing about this project is that it was a mashup between the microbial ecology experts at Arizona State University, led by Rosie Krajmalnik-Brown, and Advent Health's experts in metabolism, nutrition, and human physiology, led by Stephen Smith. So we knew that putting these fields together could help us determine whether the gut microbiota and its bioenergetics, or how it uses and redistributes energy, quantitatively change our energy balance. If the gut microbes are likely to be causal, they need to alter energy balance because that's the only way you're going to change body weight. So to do this, now we're getting into these design elements that I refer to that are critical for having confidence in our results and how this all ties into metabolites. So we had one goal, and that was in a tightly controlled manner, modulate the gut microbiota within individuals. So the only way to do this is to use what's called a randomized crossover design. So this means that you use your disruptor. In this case, we use diet as a way to change the microbiota as much as possible. We had a control Western diet and a microbiome enhancer diet that we considered the intervention. All participants received both diets in random order. And the key differences between the diets were 4 drivers. The Western diet was low in fiber, low in resistant starch, which are foods like beans and certain ancient grains that have a different structure of the starch molecule that is a type of fiber, but it's processed differently by the microbes. Lots of processed foods on the Western diet and small food particles, meaning juice instead of whole fruit. So basically, the microbiome enhancer diet was a high fiber, whole foods diet. We phenotyped participants very deeply, and I'll tell you more about some of our phenotyping methods, but here's a couple of key things. We made all the food very precisely in a metabolic kitchen. Participants ate the food 11 days at home and 11 days while domiciled in our metabolic ward. So we knew for sure that they were consuming the diet, and that is really, really a big distinction from other studies. At the mouth, both diet had the same amount of calories, and they were matched to precisely measured energy expenditure. We titrated diets to maintain energy balance, and then we kept macronutrients the same. Percent protein, percent fat, percent carbs, exactly the same. So the biggest changes were these drivers. And then all our assessments were at the end of each diet period. A couple of key phenotypic innovations were the use of whole room calorimetry, which is a way to very precisely measure energy expenditure, substrate oxidation. And in our lab, for the first time in humans, we actually also measured 24-hour methane production at part per billion resolution. Fecal energy loss was one of the mechanisms we believed the microbes might shift energy balance. And so to really understand fecal energy loss, precision is key. So we implemented a new method to increase our precision and make sure that we were comparing exactly 24 hours of fecal energy between participants. We also phenotyped body composition. We precisely knew energy intake. Gut transit time was measured with a radiotransmitter pill. We measured a whole panel of enteroendocrine hormones. We assessed hunger and satiety. A really cool aspect is that Arizona State built a model at the submission of the grant that helped to start predicting some of the microbial contributions to energy balance. And this model is continually being refined, and you'll see more publications in this space. But everything we know about a calorie today on the food label does not account for the role of the microbes. And this is some way where there could be variability in how people process what they eat. We also profiled microbes, we measured metabolomics, and have a really nice biobank. So with all that, now you can hopefully sense the level of control and precision that we so intentionally put into the study. We had a small sample size, 17 people, healthy, young, normal weight stable. So why didn't we go straight for people with obesity, for example? Because we wanted to establish the paradigm in healthy individuals first, and then apply it to other disease states in the future. So our primary endpoint was host metabolizable energy, which is essentially the energy available to us from diet after accounting for fecal energy loss. Now, there are other ways that energy is lost, such as gases and a little bit in urine, but fecal energy loss is the primary differentiator between the energy that goes in to the mouth versus what we actually see. And so we hypothesized that on a microbiome enhancer diet, the green dots, there would be lower metabolizable energy. And as you can see, it was lower by about 6%. And the other thing that really intrigued us was that on the Western diet, as expected, because all the foods are highly digestible, vast majority is going to go in the small intestine. When you feed people's microbes, which are highly variable, there's a pretty big spread in how much energy people actually absorb. Some look more like they were still on a Western diet, while others really ramped down the amount of energy that they absorbed. But what does this have to do with calories? Because that's the metric we all care about. You know, how can I reprogram my microbes so that I can eat more? That would be a great thing to do. So on the microbiome enhancer diet, people absorbed an average of 116 fewer calories per day. And this doesn't sound like a lot, but if you consistently feed your microbes for a year, that's 42,000 calories of negative energy balance. And this will not compare at all to the types of effects you would get from the current anti-obesity medications as far as food intake, but it can certainly potentially help to provide a better metabolic environment for success with weight loss interventions. So some of you, if you're nutrition people, you might be thinking, well, this could just be about diet and have nothing to do with microbes. And it's just, you know, the unabsorbed food that we're talking about here. So I'll share just two pieces of data that suggest that the microbes have an impact. The first panel you see on the left is a surrogate of microbial biomass, which is essentially the size of the microbial community. And on the microbiome enhancer diet, energy from the diet was used to expand biomass to the tune of about 8-fold higher, because this is on a log scale. So that suggests that some of the energy we could have simply reabsorbed was used to grow biomass, and energy can't be created or destroyed. So if the microbes used it, that means we didn't have access to it. The other piece is that the microbes are very different by diet. On the right-hand side is beta diversity, which shows you sort of how the community separate, and you see very nice separation by diet. And we have other microbial pieces of data that I'm happy to share during the Q&A. So that is why we should care about this, what we did, and our top line results. And I'm going to pause there and hand the stage back over to Heino, and then we'll get into the second part afterwards. Okay, so I hope from the first part of the talk, you got a sense of the crucial impact of diet on both host energy balance and microbial composition. So now we're going to talk a little bit about some of the metabolomics insights we have. I'll share some results and then really think through what are some of the methods consideration as far as the study design itself, some of the analytical considerations that we've thought through, and then what I think are some intriguing potential future directions. So I think the overall theme that I want to leave you with today is that a robust study design can yield robust phenotypes. And in our case, we had a robust microbial phenotype and a robust host phenotype. So that level of robustness leads to an opportunity to explore metabolites as biomarkers and mechanistic links. And I say this because when you have a level of deep phenotyping and precision that gives you a lot of confidence and a lot of similar directionality in the way the phenotypes change, it allows for, at minimum, asking yourself, what kind of metabolomic signature could reveal a starved microbiota on a Western diet or a well-fed microbiota on a microbiome enhancer diet? And how does that then align with clinical phenotypes? Beyond that, there's an opportunity to think more deeply about a particular microbial metabolite that could then act on a specific receptor and lead to specific release of a hormone that maybe talks to a vagal output that impacts appetite. So it opens up a lot of doors, and I think the way the study is designed is critical for this. So if you're thinking about looking at either targeted or untargeted metabolites, it's really all about what's your research question, your primary research question. and figuring out how to best design your study to focus on that primary question and not deter from that question by measuring too many things that could impact the power you have to detect your primary endpoint. So I think it's pretty clear, and it'll become clear in just a moment, that diet is critically important for understanding metabolites. Even if we're talking about just fasting metabolites, there's still a long term impact of dietary habits on the production of metabolites. So, assessing diet is a step in the right direction. There are validated tools to do that. There are more contemporary tools that use elements like food photography to really, if you're not able to prescribe a diet or provide a diet, to have a really good sense of what your study participants are consuming. Our expertise here is controlled feeding studies, and those then add a whole nother layer of the ability to know for sure that a dietary intervention was adhered to. Then it's about what type of clinical trial design do you need? In our case, we knew a randomized crossover was critical because of the inter-individual variability of both the gut microbiota and the biological factors that the human brings with them to the study. But there may be other instances where a parallel design is what you need. For example, if you want to look at caloric restriction, a randomized crossover doesn't really work because you'd have to calorically restrict, lose weight, gain the weight back, and do it again. And the impact of the prior weight loss on the subsequent weight loss would be really hard to wash out. So there isn't one perfect design, it's based on your study question. Deep phenotyping is super important so that you get a really good sense, for instance, in our case, of the entire energy balance equation: intake, expenditure, fecal energy loss, and loss via methane production from the microbiota. But beyond that, adding elements of rigor and reproducibility, so measuring the same thing in a couple of different ways if that's accessible, so that you can have more confidence in the results. And I think where we can all innovate a little bit more, particularly in the field of nutrition, is finding methods that increase the precision of our measurements. And we already had that established with calorimetry, but then in collaboration with Arizona State University, really developed a way to have a precise measure of fecal energy loss that we believe reduces the noise from assay variability and allows us to hopefully truly see inter-individual variability because of biology. So our approach is an approach you may want to think about for a clinical study. If metabolomics is not your primary endpoint, it's a secondary or exploratory endpoint, then starting with targeted, if you have a hypothesis, could be a really good way to go. And that's what we did. We knew from Arizona State's model and from the literature that short chain fatty acids were going to be a key indicator of altered microbial bioenergetics that could influence host energy absorption. So those are the results that are published and I'll focus on today. But then after we had our top line results finalized, then we decided to do untargeted metabolomics in feces, urine, and serum, and 2 serum time points, actually. And then we did lipidomics also on serum at 2 time points. So I'm not going to be able to give you very many results on number 2 and 3 because the analysis and the QC of the analysis is still pending, but I'll give you some hints that I think are pretty exciting, and hopefully you'll keep up with our group and take a look at our next paper when it comes out later this year. So I gave you 2 pieces of evidence earlier about how the effects we're seeing on energy balance weren't just about diet. And 2 of those were related to the gut microbiota, biomass, and diversity. The third one was its metabolic output. And short chain fatty acids are the primary microbial byproducts of fermentation of dietary components, particularly soluble fiber, insoluble fiber, and resistant starch. And so you can see from this panel on the left, or this only panel I'm showing you now, that in the green dots, the microbiome enhancer diet, the short chain, all the short chains that we published so far were higher. So total, which is a combination of the three main short chain fatty acids, acetate, propionate, and butyrate. This is on a log scale, so it looks like a small difference, but it's actually pretty robust. And one thing I want to point to is that we assess fecal short chain fatty acids as micrograms per day. A lot of studies do micrograms per gram, but in our case, we knew that with a microbiome enhancer diet, there should be a much larger fecal amount. And because we knew exactly how to adjust to 24 hours of feces produced for all participants equivalently, we were able to extrapolate to micrograms per day. And I think this is one important advantage because especially with fecal samples, they're not collected at the same time of the day. And there might be some variation in what you see per gram. But when you look at it per 24 hours, you get a better sense. What's really needed is a better understanding of flux of short chain fatty acids throughout the day to truly get to a more precise quantitation of each individual's total production. Not to mention that flux is also very important. So not just what's made, but how it's utilized. The other tissue we measured a targeted short chain fatty acid platform in was serum. And you can see that for all short chains, except for propionate, they were higher on the microbiome enhancer diet. So we have a pretty good amount of confidence that when you feed the microbes, they grow, and one function that's robustly upregulated is fermentation. So we are very excited about that first paper, and all these data are in that paper that is uploaded in your resources, but we wanted to go deeper. And one of the key drivers for me in doing mechanistic translational science is figuring out how to scale it up and look at the translatability to a real world setting or to big population studies. And in my mind, out of all the data we have, metabolomics could help us achieve that, because if we're able to uncover a really robust set of biomarkers for these diet host microbe interactions in a very controlled setting, then other investigators that have large population studies with different diets or other parameters that think, oh, I want to look to see what those biomarkers look like in our study and see if that helps us to understand and their dietary intake, then I think the metabolomics could really help us do that. So we've been doing a lot of thinking about how to best analyze the data, and there are many considerations that can vary by study and by preference from the investigator. First thing you want to think about is whether you want to filter out metabolites that are absent to some degree. In our case, we knew that there would be some metabolites based on our study design and the control of that design that would be very low or absent, for example, on the Western diet. But when we reprogram the microbes on the microbiome enhancer diet, those metabolites would emerge or shoot up drastically. So we knew we had a chance of biological reasons for there to be presence or absence by diet. There's also a really nice white paper that Metabolon put together about their filtering and imputation techniques that gives a lot of confidence that when metabolites in the untargeted platform are below the limit of quantitation, it's not because there's some sort of mistake in the assay or anything. It's truly because they're below the limit of quantitation. So we chose to filter by retaining metabolites that were present in greater than 75% of samples from one diet. You could filter by 50% across both diets, but we knew because we would have this dichotomy in some metabolites within diet that filtering this way helps us to maybe get rid of some metabolites that are absent purely by chance, but retain a robust set that were truly different because of biology. Then as far as the statistics, there can be some simpler tests such as a t-test or an ANOVA, but all of our data, because it's a randomized crossover, has been analyzed with a linear mix model that takes into account both the diet order and the period that was first or second. So we're able to use that model to evaluate effects of carry forward from the first diet, and also order effects. So we uh, we use that linear mix model. And by the way, as far as imputation, there's a lot of different thoughts on how to proceed with imputation, or whether to do it at all. In our analysis, we imputed with the minimum of each metabolite that was retained. So these are some high level sort of qualitative results that I can show you today that to me are very exciting. So the PCA plot on the right shows you that there's robust separation of samples based on their metabolite composition by diet. So much like the microbiota went like this, the metabolites are also quite robustly different with this controlled diet. And these are just the fecal metabolites I'm showing. Out of the fecal metabolites, we received 822 were picked up by Metabolon's platform. And after filtering, we retained 631. And so far, 158 of those metabolites are different by diet after false discovery rate correction. So I'm confident we're going to have a very nice signature that we can put in the public domain that may help others really evaluate some of the components that we evaluated in a population level study. And Daria Gudisman and my group is leading these analyses and doing a fantastic job. So keep an eye out for her publication of this with our entire group of collaborators. So what's next? What kind of interesting thoughts are we having about where we go next? These are just some ideas that we're contemplating, or actually some of them we're implementing. The first is thinking about fluxomics. So that's instead of flux of one metabolite, which could be very interesting. And we're also interested in doing, for instance, with short chain fatty acids, as I mentioned, looking at flux of multiple metabolites or all the metabolites we think is very key, because there's diurnal variations in some metabolites, and then there's the impact of diet on those metabolites throughout the day. There could be other signals that have you produce a metabolite at low levels in the morning and at high levels at night. So we think this is an important future direction for us to get a better understanding of the actual mechanisms that it just can't be uncovered with just a spot measurement. Then, as Heino mentioned earlier, there are lots of omics techniques that are very valuable: proteomics, metabolomics, epigenomics. And it takes a specialized kind of bioinformatician to develop pipelines to integrate and allow us to learn concurrently from those data sets. And we'd like to move in the future towards that integrative analysis of multiple omics platforms. Then, a lot of what I've presented and what a lot of papers present when it comes to mechanisms, particularly about the gut microbiota, rely on relative abundance changes. So within each sample you're comparing, what is the relative taxonomic composition of whatever species are present relative to one another within that sample. But that doesn't tell you necessarily that one species is specifically upregulated. The relative abundance can go up because other species go down. So really understanding the quantitative aspects of the microbiota and then how the community actually functions, we think is really key to getting to mechanisms. And we have a grant that we're hoping will get funded very soon, that we're collaborating with Karsten Zengler at UC San Diego, where he has these amazing next level pipelines of exploration of microbial community dynamics, and particularly what the microbes eat. So that the hope is that if we can figure out what the microbes eat, we can figure out what to feed the person to feed the microbes we want to feed to achieve a certain composition. So that's, further out in the future, but we have hopefully a grant that will help us get started in that direction. And then with the host side, is really thinking about other phenotypes that we can connect directly to a microbial function and or a microbial output, so that we have a clear set of steps that lead from the change in the microbiota to the phenotype that maybe can be targeted therapeutically. As I mentioned earlier, scalability is very important to me. So not a lot of centers could do the type of work we do here at the Advent Health Translational Research Institute. So we need to figure out how to come up with panels of biomarkers or pathways that can that are informative but don't require the level of study that we did to help us learn more about what's happening in global populations. And a final really big interest for us is therapeutics. So we do a lot of work with various pharmacotherapies, anti-obesity medications, diabetes medications, et cetera. And, a lot of the new generation anti-obesity medications target hormones that are impacted by the gut microbiota, like GLP-1. So could optimizing the gut microbiota improve maybe not so much the efficacy of those drugs, because the efficacy is pretty startling, but maybe improve the longevity of the effect, especially if the drug is eventually titrated down. And then a key passion of mine, and we're about to submit actually tomorrow for consideration, a review on what our thoughts are on how to develop precision nutrition's approaches to target an individual's specific microbial community to achieve a desired outcome. And I definitely know that, nutrition is a key component. And if we can even get beyond, 2 diets like we studied, but tweak those diets in very specific ways, that would really move us towards the precision nutrition avenue. So in summary, what I've covered today is, hopefully, if nothing else, if you do nothing else today, when you eat lunch after this webinar, well, depending on what time of the planet, what part of the planet you're in, maybe you're not at lunch yet, feed your gut microbes. Think about what you could change or add to your diet to feed those microbes, because when you don't feed them, they find other ways to feed themselves, like breaking down your mucus lining in the gut, which is not so good for you. Happy microbes are important for our health, and diet is a powerful modulator of the gut microbiota. I've also showed you that the gut microbiota impacts human energy balance. In our hands, in our first publication with our design, primarily via altering energy available to the human host from diet. That doesn't preclude other mechanisms from being important in situations like caloric restriction or obesity. We believe that we'll be able to show soon in a publication that metabolites can be robust biomarkers and mechanistic signals of integrated diet-host microbiota interactions. And the field would benefit from developing these mechanism-specific metabolomic signatures that can be leveraged therapeutically. And with that, I would like to thank the entire team at the Translational Research Institute, particularly Stephen Smith, for bringing me onto this project 8 years ago, our postdocs, our cores, and then the team at Arizona State University, led by Rosie Krajmalik-Brown, and the funding that was used to generate the results we've shown you today. And with that, I'd like to thank you, and you can scan that QR code if you want to have my email handy in case you have questions that you can't answer today. Thank you so much.